we were talking about this. I remember that. Did I cover all these cells? Well, if not, summary is there should be 4 different types of these glial cells. Remember that glue means glue, holds everything together. We've got astrocytes that are sort of structurally very important. You can see how they span and sort of hold things in place and hold stuff together. The previous slide discussed many facets of these astrocytes. Okay, anything from making the actual blood-brain barrier that I'm getting ready to talk about, all the way to actually helping repair brain injuries and that kind of stuff. So review that. Microglial cells are your little, very sort of frayed out kind of cells that are moving all around. Remember I said that there is a blood-brain barrier, so there's not a lot of immunoactivity with our white blood cells and the such going around in our brain tissue. So these microglial cells migrate all around and take care of that, perform that function. The oligodendrocytes, their biggest job is to make the actual myelin sheaths that wrap around brain neurons. Schwann cells, remember, do that in the peripheral nervous system. This becomes very important. Now remember, what is the most important benefit that we get from myelinated nerve fibers? What does it do for our nerve transmission? Speeds it up, incredibly so. It makes it much, much faster. So it's much more efficient. Remember that where these little gaps are in between, they can sort of skip and jump as opposed to just being straight all the way through. It can go a little bit faster that way. And then the ependymal cells will form the lining. Sort of at the surface, it'll be a separation between that tissue and, for example, the cerebrospinal fluid that's outside of that neural tissue. I'll discuss the fluid now. Okay, as far as protecting our central nervous system, obviously that's extremely important, and our body has pretty thick bones even to do so. Our cranium and some of us are thicker craniums than others perhaps, but as our vertebral column as well, and all those vertebrae. The vertebral column is pretty neat in itself, and then it's, instead of being a solid bone, it's just actually a whole bunch of rings stacked on with sort of soft spacers in between them, that cartilage tissue in between them. That will allow more flexibility for us. But in any case, you've got several layers that go around the central nervous system, around this neural tissue. We call that the meninges. Several of these meningeal layers are what we're going to talk about here. The outer is called the dura mater. The middle is the arachnoid, and the innermost is the pia. And each one is a little bit different. Now we're talking about a very small space here. A lot can go wrong, and a lot can happen in these tiny little spaces. But it's a very, very thin, to the point where if you were just picking it up and looking at it like a brain that you had preserved, you wouldn't notice this. Okay, this is the microscopic differences between these things. But nonetheless, they are layered, and there are 3 different areas, and I'm going to show you where these are. Okay? Now, around all of this, and actually within what's called the subarachnoid space, we'll talk about that in a second. So sub means below, right? So here's the innermost, and then outside of that would be this. Okay, the innermost is the pia. Outside of that would be the arachnoid, and then on the very outside is the dura. Okay? Actually, between these two, between the pia and the arachnoid, is where some of this fluid is going to start circulating. Okay? And it's going to interplay with the blood-brain barrier. But in any case, we have the cerebrospinal fluid. Now, this special fluid that actually circulates within the tracts of our vertebral column, around our brain. It's very important. The blood-brain barrier ends up limiting the access of the blood-borne substances in the brain. Okay? Now that's good and bad, of course. Certainly bad things, pathogens and the like, it makes it much more difficult for them to leave the blood vessels and get into the brain. But at the same time, things that we need to get out of the blood also are more highly controlled. So we have coping mechanisms for this. Okay, just sort of in summary here, if you take a look at this picture, these numbers sort of represent the path of the cerebrospinal fluid and where it's traveling. It's actually made in the choroid plexus. Actually, 2 different ones. I think the book calls them plexuses, but you could also call them plexi, I suppose, but plural form of plexus. If you look sort of in the center, in this third ventricle region especially, in the brain, is where there's specialized cells that are going to produce and secrete the cerebral spinal fluid. Okay? And it has to be constantly secreted and produced because it's not recirculated as well like the blood would be. It's sort of produced and it ends up mixing with the blood is what ends up happening. So it's sort of constantly produced and actually makes its way out. And right here at the beginning, what's important to note is that you can see that it's still actually inside that neural tissue. It's sort of inside the brain itself. And it travels down towards through the brain stem, or towards the brain stem, I guess. And notice where it says number 3. I guess you can see all of them down here. But it says it exits the 4th ventricle at the base of the brain. So at the base of the brain, it actually moves from the fluid that has been produced from the sort of the inside of the brain, moves through that tissue and reaches what we call the outside of the brain. Okay? Now you can see, I mean, that's going to be very important that we can have a passageway from in and out, okay, because there has to be communication there. But now this is what these sort of arrows going around right here means. It's sort of circulating around. Now it can reach the outside of that tissue. And once it's on the outside of that tissue, it starts to fill what's called the subarachnoid space. Now let's look over here. Notice I said those 3 layers, right? The different matter layers: pia, arachnoid, and dura. You can see where the pia is the very inside here. Now these dips, these are the convolutions, the wrinkles on your brain. Okay, so it dips way down in, even deeper than sometimes what you see here. So you've got the pia mater right here. You've got the arachnoid right here, that red line I can't reach because I'm too short. But and then the dura is actually a little bit thicker hide right on the outside of that. So you've got sort of a brown line, a red line, and then that sort of peachy looking stuff on the outside of it on this picture. What's very important is, notice arachnoid is, you think of spiders, arachnids, that kind of thing. You've got this web type of spidery type material that actually is filled with vascular tissue sort of in there. Branches in here. Now it doesn't go branching down in, capillaries don't fill the brain tissue. But what you have is this space right here, and this space that you see here, this spongy looking space right above the brain, but underneath this layer, it's a very, very small layer, a very, very thin space, you've got this cerebral spinal fluid circulating, and you can see it right here coming in that blue arrow. Okay, it's circulating through there. So what you get is a transfer of materials from the cerebral spinal fluid and the blood right around in this area. Okay, this is where it occurs. Okay, at least for the most part. All right, then what will happen is, that those contents will sort of mix in with the blood, and they'll switch whatever they need to do. And this continues to produce more. This continues to circulate. Okay, and this goes all the way down. Notice we've cut this out. This is the spinal cord. This is the bottom of it. This cerebral spinal fluid actually goes the whole length of your spinal cord. Anywhere the central nervous system is where you're going to find this stuff. Okay. Now we're talking about a very good, highly selective barrier here between the actual tissue of your central nervous system and your blood supply, for obvious reasons. Okay, now the only exception to this, we can say that blood does not come in contact with brain tissue. Okay? However, the exception to that would be the hypothalamus, and sort of the thalamus-hypothalamus region. Now, I'm going to talk about these guys in a second, but what you probably have at least known up to this point is that is sort of the control center of the brain in terms of homeostasis and in terms of the physiology of the body. How you respond, what your body does to offset levels and that kind of thing. The hypothalamus is the sort of the command center for this. For a simplified explanation, you can watch Osmosis Jones. You can see that the headquarters for the police station of the white blood cells there are actually the hypothalamus. But anyways, now one thing to note about the brain, okay, or about the central nervous system in general and the cells within it, but obviously most importantly the brain. It needs a constant input of oxygen. Our other tissue in our body can adjust. In order to make ATP, however, in the brain, we have to have constant influx of oxygen. That's why whenever your oxygen supplies are cut off, not only do you have to have oxygen, but you have to have a constant stream of glucose for the brain. Other body tissues have the ability to sort of store things, and then when there's a shortage, use them. A muscle tissue especially can do this. The brain cannot. So generally the first things that start being affected, like when you get low blood sugar or when hypoxic situations start to occur, the first visible manifestations in this patient is going to be something cerebral. like whether it's vision starts to fade, you start to feel lightheaded, something associated with the central nervous system usually is one of the first things that are affected. And it's because of this need. There is no anaerobic metabolism. We talked about back when we did cellular metabolism, we said pyruvic acid was forming, right? And we said that when in the cytoplasm, if pyruvic acid formed, then it could go and split into acetyl-CoA and go into the whole Krebs cycle thing. However, if there was no oxygen to function on that electron transport chain, then pyruvic acid would be made and everything gets backed up because it can't get in the Krebs cycle because there's no oxygen there to accept those electrons. So what happens is that pyruvic acid can get converted into lactic acid, but anytime that happens, there's still like a couple ATP that were made and all that kind of stuff. Well, that stuff doesn't happen here. Okay? That's why we have a very important need for a constant input of oxygen and glucose to the brain. Okay. So there's a sort of a perforated type of system that you would get normally with vasculature in our body. The cells that wrap up, our blood vessels are made of cells that are wrapped up cells that make these tubes. Usually there's small enough pores for things to migrate in and out. However, in the case of the brain, you can see it's much more tightly held together. You actually have tight junctions, almost like these are stapled together, which is very different than the rest of the body and the blood vessels in the rest of the body. And not only that, you have these astrocyte processes that are feeding in and sealing up all around all these brain capillaries. Okay. All right. So, very tight barrier. Come on, do this too. Starts to spin around. Okay. Now we know this to be true. I shouldn't have to lecture on this. Different ways to hurt your brain. Okay. So obviously the first two. Now swelling and hemorrhaging obviously are very important in the brain, mostly because there's not a lot of space to alleviate this type of pressure that occurs. If there's swelling, if there's any kind of tissue edema in the brain, it can compress, and there's a lot of neurons associated with that. There's a whole multitude of things that could go wrong whenever you're dealing with the compression and interference of neural activity. So that's obviously why it's a high roller type of game when you start gambling up there. But a CVA, a cerebral vascular accident, fancy word for a stroke, you guys probably already know that. But keep in mind what a stroke can actually be caused of. It's any time that O2 is deprived. The brain tissue is being deprived of oxygen. That could be a rupture, say resulting from some type of aneurysm or some sort of vascular tear, anything like that causes a rupture that would obviously prevent the oxygen from flowing to where it goes. That's one way. A clot could clog up the system, preventing blood from flow and thus oxygen being delivered where it needs to go. Another way, one of the very, very important reasons to get air bubbles out of things, right? You don't want to shoot air bubbles into bloodstream. An air bubble can act just like a clot. You guys probably already know that, right? The bubble can block the flow of blood. So not only oxygen and glucose, right? So not only oxygen, but also glucose. Both of which obviously we're going to have a huge impact, especially on neural tissue. Okay, one thing though that, I don't know, you may not know, is why is it, if someone has a stroke, some things are destroyed. I told you that neurons generally cannot regenerate, like brain cells and that kind of stuff. And then you start thinking about, and I mentioned this before, but you start thinking about people who had strokes and they've lost the ability to speak and they have to learn to speak again. How do they do this? Well, they actually form new neural connections is what they do, especially people who literally have to relearn this. And we'll talk about what learning is and memory is here in a second. To literally have to relearn something means you have to form new synaptic connections between different cells. And you're obviously not going to be able to use the same cells that were there. You're not going to be able to create the exact same pathway that was there. But you can take a different way to get there. Okay? Now, also, along those lines, why is it that the effects of stroke can sort of depend upon the time between the onset of the stroke and the treatment of the symptoms of that stroke, right? Now, we know that there's a difference there. Generally, what's most important to keep in mind out of this is this next statement. It says the release of toxic glutamate from these damaged cells overexcites and destroys. Now, what I mean by that is, you could have a blood supply and a stroke in a very small amount of area that affects a very small region of the brain. But yet, it may actually that stroke may end up affecting a larger area than just that one place where that would have been fed by that little vessel or whatever. And the reason why getting to it as soon as you can, for example, and preventative type of healthcare and that kind of stuff, the reason that you want to do this is because what has been shown to happen is cells that are damaged by stroke, they're not receiving their oxygen, not receiving their glucose, and thus they die. Neural cells tend to release glutamate, which is normally there, but supposed to be controlled within the cell. And when they start to die, it starts to be released. And what happens is the glutamate from the damaged cells get released to the cells that normally were not damaged. And up until that point, we're functioning. When they start taking this in, it actually overexcites those neurons. It's sort of like acting like a neurotransmitter here. It'll overexcite those neurons, almost just causing them basically to sort of spaz out and ultimately destroy those neurons, where normally maybe if that could have been treated and that glutamate prevented from spreading, you may not have had the severity of that stroke. Okay? You'll also see, and this is where some of the medication is going, whenever people are coming in, some of the attempt to help this person who suffered from a stroke is not immediately preventing this from spreading, for example, trying to slow down the release and destruction of these cells so that they don't destroy other cells that previously themselves were not destroyed by that stroke directly. All right, infections obviously are of extremely importance due to the fact that we have such a tight blood-brain barrier that applies to our immune system as well. So infections are of grave concern in the central nervous system. Of course, brain tumors, we don't need to talk too much about all that. Okay, now, to mention stuff here, I don't want to get too much into the anatomy, but I have to mention some, because what I want to point out here is the way the anatomy is related to the function of those parts. I'll show you what I mean. What's really interesting is if you look at comparative biology, and you look at what we consider primitive organisms and more complex organisms like us, like our cerebrum, the big cognitive part of our brain at the very top is much larger proportionally, percentage-wise, than the rest of our brain compared to these lower organisms. So what is the physiological explanation in relationship to that? Because what you'll see is sort of actually physically the lower on the brain the part of the brain is, the more primitive its function, the more basic and essential it is to surviving life. Okay? Like your brainstem is the very bottom, actually before you even get into what we would maybe call the brain, the actual brainstem itself. It is responsible for things you don't think about. It is responsible for life-sustaining activities, your respiratory rate, your cardiac rate, basic things that happen all the time that we don't concern ourselves with. And not only that, I'm going to show you a picture a little bit later. I don't know if you had to memorize all the cranial nerves and all that kind of stuff, but you don't have to in this class. But notice how many nerves branch out from the brainstem. Okay? It's a really an important hub, a central point between like the spinal cord and the brain. From just raw processing and total sensation from everything that around you to process to a cognitive level to make sense of all that sensation. There's a central hub that has to be passed through, and that's the brainstem, so it's incredibly important. Okay? Now, the cerebellum sits right behind that, and we don't use it a lot. We don't use it for thinking, but we do use it for motor movements. It coordinates a lot of the neural transmissions along with the muscles that are attached to them, so that it's sort of why we can stand up and not fall down. I don't know if you appreciate that. You don't really think about that, but the fact that you can stand up on two legs greatly separates us from almost every other animal that's out there. How is it that I can just walk like this, and have that much muscle coordination in opposite directions to hold myself upright? So the cerebellum integrates all of that. It also is the reason, like when you learn how to ride a bike, you don't forget to learn how to ride a bike. Okay? Once these pathways have been established, it sort of remembers that. Not necessarily at a cognitive level, you don't have to think about it, but your motor movements form sort of like motor memories in the cerebellum. All right, now above that, I'm going to show you pictures of all this, but is the diencephalon, which is comprised of two big things I want you to know, and that's the thalamus and the hypothalamus. Hypo means below, right? So the hypothalamus is actually a little bit anterior, or actually ventral, a little bit ventral and below the thalamus. So just in general, of the two, the hypothalamus is extremely important for homeostasis, controlling thirst intake, all that kind of stuff. We'll get into all that with endocrine system when we study hormones and the like. So now we're sort of moving up now, is what my point is. The brainstem is sort of like, things that are basic to life that you don't have to necessarily think about, and they're just happening. On top of that, now you're increasing that complexity of this organism, and not only are you able to do all this stuff, but now you're able to body position, control your body position, move, get through your environment, which is a step up. Even further than that, you can in a more sophisticated fashion with the diencephalon, a higher region of the brain, control homeostasis, respond to a wider range of fluctuation in your environment and to live under these conditions. And then obviously the most complex would be the cerebrum, where all this is integrated. You've got your gray matter and your white matter, all these different types of cells. Gray matter and white matter, if you remember I mentioned this before, that's okay. If not, it's up here. Gray matter consists of mostly cell body types of regions, okay? And your white matter is going to consist of mostly axonal regions, actual fibers. The reason they're sort of whitish in color is because these cells that are myelinating these are heavy in lipids. So you get sort of what lipids would look like at a microscopic level, with sort of a whitish color. Okay, and I mentioned this already too. The basal nuclei is a special region of the brain that's sort of down in the center. And what's unique about it is that in this sea of white matter, most of your gray matter, most of your cell bodies in your brain are actually up at the top, where all those convolutions are and all of those folds. They're firing, and as they go down towards the center of your brain is where they're all branching off and sort of connecting onto each other. Okay? Now what's interesting about the basal nuclei is down there within all of that are these cell bodies. So they make gray matter in the sea of white matter. And as far as what they do, I've sort of already mentioned that, but they are sort of important especially for getting rid of extra movements that aren't necessary. Movements that normally would be happening all the time because of all the neurons that are firing, they sort of weed those out. Okay? And that's sort of the symptoms of Parkinson's disease. That's what I mentioned up here. The basal cell, so the basal nuclei are affected. The neurotransmitter in that region is affected, and as such, they don't function like they should. So you get a lot of extra movements and shaking and all that kind of stuff. Your cerebrum gives you consciousness. You could philosophically digress too many points here, but we'll stick with the biology, I suppose. It allows you to realize that which you are experiencing. It's possible that a lot of things that are out there experiencing things, a lot of animals even, that we would classify as animals. You realize that coral is actually classified as a lot of people think it's a plant. Some things cannot perceive consciousness. We can. So you get a complex and larger types of things on top of simpler and smaller things in the brain. Estimated to be about 100 billion neurons. So, those are things that are actually firing. These are cells that have electrical potentials associated with it. All right, there's a picture of all these things. So your brain stem's down here, very primitive. Cerebellum's also very primitive, but separate from. It's got a different function. Here's your diencephalon, which is the thalamus and the hypothalamus. Notice here something very interesting I'll refer to a little bit later. You probably mentioned it in basic anatomy, but something called, excuse me, the limbic system, right? Well, all this is going to be tied in. This isn't a picture of the limbic system, but there's going to be a big chunk of this associated with it. The limbic system also involves some stuff coming up here, your olfactory bulbs and all that. But here's a basal nuclei, this traced outline here. So it's sort of a gray area in the center. Okay. You've probably heard this before too, right? If you're left-brained or right-brained. They do have some, on average. Now, this is not perfect in every person, okay? But on average, what tends to happen is the cerebral hemispheres take on specializations. And yes, it's true that sort of the left side of your brain controls most of the right side of your body and all that. And I'll show you an example of this in a minute. But as far as just thinking and cognitive thoughts, not sure why this is the case, but what it tends to happen is the left cerebral hemisphere excels in performing logical, analytical, mathematical types, verbal tasks, very systematic, very logical approaches to things. So someone who is very left-brained is someone who's probably very organized, a very logical thinker, very sequential, sort of make lists and check them off, that kind of stuff. Touching on any nerves right now? So you know, you might go into their office and everything might be kind of like this, sort of like this, and maybe a little bit like this. I don't know. Call me left-brained. It's fine. Now, the right cerebral hemisphere excels in spatial perception, artistic, musical talents. I see one as organized and one as chaos, sort of, is my version of this. The right cerebral hemisphere, spatial, I mean things like you can imagine where things are, like in space. I don't mean outer space, just like everything out here. You can imagine where things are relative to one another. You get a good idea of what something's going to look like before it happens, kind of thing. That's a good spatial perception. People who are, artists, people who can draw, paint, contractor, architects, those kind of people who can sort of foresee all of this and imagine it before it ever is done, are very strong at this type of stuff. That's a good spatial sense. Musical talents. So, I don't know. You can classify yourself. I'd say I'm about halfway in between these two, actually, right in the middle. So, not for the drawing part, though. Yeah, I'm over here with this. Okay. The EEG, electroencephalogram. Now, realize that when you're looking at an EEG, you're not looking at somebody thinking necessarily. You're looking at just postsynaptic firing in general. What kind of neural activity is happening? What kind of potentials are being sent? Just after a synapse. Now, this is going to you stick electrodes all over people and that kind of stuff. Sort of like an EKG with a heart. You realize it's not actually corresponding to a heartbeat. All it is measuring is potential changes from one area to the next. When a potential happens, it sort of can spread through the fluid. It can spread through the water. It can spread through the cerebrospinal fluid. We have this electric charge spreading through us all the time at a very small level. And this is sort of, these sort of diodes everywhere that are measuring this. That's what they're measuring. This stuff is, this potential that's being discharged throughout the tissue fluid and that kind of thing. Now, as far as what it can be used for, well, mostly just cranial activity. What is happening? Is anything happening? You can get into some pretty hairy issues with this, but it can be used as a clinical tool to diagnose cerebral dysfunction. Mostly, sort of, is something spastic happening? Is something sort of intermittently happening? You can't tell exactly what is happening because all you see is a bunch of waves like this. Unlike the heart, where you can sort of the PQRST and you can sort of assign certain areas to certain things. You can't really do that with an EEG. But nonetheless, what you can do is, and I'll show you how, it can distinguish various sleep stages. Sleep is a very interesting thing to talk about. Legal determination of brain death, but of course then, that's, like I said, it's hairy with that, but that's some of the applications of it. Something interesting, just keep this in mind. There are two different waves on an EEG. There's what's called alpha and beta waves, just so you know what those are. Notice it says eyes closed, and you've got sort of these bigger waves that are a little sporadic. Call those alpha waves. You open your eyes. Notice what happens. You would think sort of the opposite would be true. You would think sort of the more your eyes are open, the more your senses are acting, the more neural activity that would be going on. But interestingly, the opposite is quite true. And you see this. So close the eyes again. You're back to where you were. Okay? Now keep this in mind as I go through in a second when I talk about sleep. Okay. I mentioned the limbic system. The limbic system is no separate component. You can't look inside a brain, cut it open, and see the limbic system there. The limbic system is just an area of cells. It's largely undefined. It's not like you can go in there and trace it. It's just these areas that seem to constantly work in the same way within people. Okay? And you'll also see that in the brain when you start studying brain anatomy, it's incredibly complex because there's parts within each of these regions that span out and affect different regions, and then you got to redefine all the boundaries of all this stuff, and it gets crazy. But one of the things we can say about the limbic system is that it's very important in your emotions. It takes your inputs and your experiences that you are feeling, your actual neural impulses that are being sent, and integrates them in a way to where you associate them with memories and you associate them with emotional behavior. Now, there's a lot to that. We can't explain how or why, but that's all that we can say at this point, is there's a relationship between this area and your emotions. So as such, this limbic system, sort of in the center of your brain, it will work with the higher cerebral cortex to control behavioral patterns. So that means it makes you aware of it, and you can sort of make decisions based upon your emotions. It sort of has reward and punishment type centers. In other words, you know that this is good and you like it, so you keep doing it. That affects your behavior. You know that this is bad. You don't like it, so that affects your behavior. That's emotionally tied to the receptive inputs that you are receiving from a neural perspective. Okay? So you integrate that all together, and how that affects your behavior, what you feel is good and what you feel is bad in terms of the choice that you make as a result of that, largely is affected by this area of the brain. You start screwing with it, you start really screwing with people. All right. Now, here's something interesting. For example, the neurotransmitters in the pathways here that we're talking about for emotional behavior include serotonin, norepinephrine. Okay? So you start thinking about like medication for depression and that kind of stuff. How do these things work? Or illegal drugs. How do they make you feel better? How do they make you feel worse or whatever? In the case of this, in the drug Prozac, can take these neurotransmitters. In depression, there's actually a deficiency of this or pair or single neurotransmitter. So as such, it would make sense that if there's a deficiency of the neurotransmitter present, there's not as much neuronal firing happening. There's not as many impulses being sent. So the things that are associated with positive emotion aren't, right? I mean, it's not being sent. There's a lower level of happiness maybe associated with this. So there certainly is a chemical tie with this. No one would deny that. At least, of any science background, no one would really deny what's going on here. What Prozac can do is actually block the reuptake of this. So in other words, what it can do is interact with the receptors that take back in this neurotransmitter. Because remember I said that those things have to be uptaken very quickly. So when they're dumped out, they get sucked back in pretty quickly. What Prozac can do, for example, is to keep it out there longer. Leave it there in the synaptic gap, and the more of it that's there, the more of it is there to interact and continue to create more impulses and actually physiologically change the emotional state of what's going on there. Now, you can decide whether that's good or bad. The issue is not, is there any physiological change going on? Because the answer to that is yes. The issue becomes, how much physiological change would warrant a medicated condition? Okay? And that's where when you get into that area of science and you switch over from pure biology into psychology, which is a softer science. I'm not saying it's bad or weak. I'm just saying it's what's called a softer science. It's a little bit less rigid. It's a little more interpretive than what you would have with biology. There's a lot of conflict there. There's a lot of differing opinions as to how that would work. In the case of schizophrenia, you actually have the dopamine. There's several neurotransmitters that are associated with the limbic system. But take dopamine, for example. A person with schizophrenia has too much. Now, I'm not sure. Maybe you could look this up on your own. I don't know why there's too much. I don't know if it's because there's a problem reuptaking it. I don't know if it's a hypersecretion of the neurotransmitter. Somehow, what ends up happening is in that synaptic gap, there ends up being too much neurotransmitter present. And as a result, you get an overfiring. So their mind's going crazy. There's way too much neural input happening in this person's head, which leads to the obvious symptoms that we all know of when we think of schizophrenia. All right. So, if you can figure that out as far as medication, we tease, and I'm guilty of it too, but in all seriousness, I mean, this is a medicated type condition to say, did you take your medicine today? Well, let's be serious, because, I mean, that could make a whole difference in what's going on here. If you can increase the reuptake of that and pull that out of that synaptic gap, it's going to make a physiological difference in that person's behavior, without doubt. I mean, we don't really have, there's some things we don't have control of. And, how our neurotransmitters work, we don't have control of. So. All right. There's a shot of some of the regions that work together to form the limbic system. Now, that's not an excuse to start acting crazy in this class and you just blame it on your neurotransmitters. It's not going to cut it. I don't have any medical documentation for any of y'all right now. So, but notice here, what feeds into this emotional area, this limbic system, is various reasons. We don't have to go through all these names, but I will point out the olfactory bulbs here, your smell. You have a lot of things that associate. You can actually get happy or get sad. You don't really think of it like that, but certainly can affect your emotions. The taste of things, the smell of things, all that kind of stuff. Okay? For example, I hate the smell of candles. It actually infuriates me to the point now where I can't take it anymore. My wife goes in those stinking stores, and I hate them. Well, I just hate candles. I don't know. It's dumb. They're always she's into this the little hot plate that melts the candle. They're sitting around our house. It's just dumb. And then I go to bed at night. I'm shutting off all the lights, and I see these little glowing lights everywhere because she forgot to shut off the candles. So she's in bed, and I can't say out of spite not turn them off because then the house burns down in the morning. And then what have I proven? Nothing. So I got to get up and I got to go turn them all off. Well, that's ridiculous. I mean, I built that house. All right. Anyways. Camera's on. I'll answer it after the camera's off. No, I don't know. I mean, the house is new. It doesn't stink yet. So. Well, that's outside. That's irregardless. That has nothing to do. That has nothing to do with what we're talking about. No, you don't want... All right, enough. All right, memory. This memory is something actually very interesting. Memory is laid down in stages. So there actually is very much truth to the whole, the statement, practice makes perfect, keep practicing, whatever. Because when you lay these memory stages down, at first they're transient type of connections. They start to form. A memory trace actually is nothing more, and this is weird to think about. It's nothing more than just a new synapse that forms. Okay, a new synapse starts to form, and you integrate, who knows how many cells into that connection, and now you've got this new path that is developed. You've got so many synapses so close to other synapses. Sometimes things get screwed up like that. They actually sort of activate each other, and you get these nervous tics and weird stuff. But a new synapse can actually start migrating, and the more you think about it, the more you're directing it, can be attracted to another place, and all of a sudden you form these connections with nearby synapses, and you integrate new information in doing that. And that's really weird. It's not like it's these words and these files that you're just storing in your head. It's not like that. I mean, it sort of is, but biologically it's not. In concept it is, but what is actually happening is nothing more than this synapse now creating a new this axon creating a new synapse with this dendrite over here, and bam, you got it. And then all of a sudden, you've got a new memory trace. And that's freaky to think about. But anyways, so to define it here, you can define memory yourself, the acquired knowledge for later recall. Now, we know, even as you know, studying for these tests, you have short-term memory and you have long-term memory. Okay? A memory trace, like I said, it's any neural change responsible for the retention of or the storage of this knowledge. Now, these traces are going to be present in multiple areas of the brain. It's going to, you got to realize that you cannot assimilate new information. You cannot form a memory of something without prior experience. Think about that. Okay? Because what happens is, if you're going to form a memory for something, the only way that you can define what that memory is by using things that you already know. Okay? That means there already has to be these synaptic connections present that you are aware of in your brain that you can then use to redirect and reform to make this new memory. Am I making any sense? At least, even if it's an abstract type of sense. So this is really starts thinking about things. What do you have to start with? What is the capacity of the brain when you're born? What connections are present? Are there any actual memories that are there? What are instincts? These kind of things. It's really weird to think about. But anyhow, some interesting points here that should help you study. Okay? Concepts and images are what are stored in your brain, not words. Okay? Not words. Not verbatim information. This is why, if you are trying to memorize things, as you study this and you try to look at words and you memorize words and you memorize definitions of words, and that's all you're memorizing, that will continue to stay in your short-term memory. It will not stay in your long-term memory. And the reason is because, think about the memories that you have that are not gone. Think about them. And what you have is a visual description in your head of those memories. Okay? You do, at some level. Whether it's a trip that you took, whether it's a concept that we have learned in here, whether it's the potentials on either side of the cell membrane, whether it's the birth of your first child. What you don't have in your head is a word that says birth of Tom, November. You don't have this. What you have are experiences that are synthesized from all of your senses and produce this image. You can close your eyes and still have that same image as if they were open right now as you think about that. Okay? So what you store in your long-term memory, in the experiences that you keep with you, are things, they're sort of images and concepts. So how that relates to you, hopefully, maybe some of you are able to pick this up. And I witnessed the difference of this whenever I was a student, is when you're starting out and you're taking these first 2 years, you're learning how to study. Now, for some of you, that's it, and you're done. For me, it wasn't. And that's okay. I'm just saying it's a difference. And fortunately, at some point, I realized that what I need to be able to do is to look at this information, and as opposed to try to memorize it, understand it. And what that means to me is whatever I need to do to try to understand it. I have an ability that, I don't know, maybe a lot of people have, maybe we all have it. I just found out how to use it, but I have the ability to take something like this and bring words on a page, off the page, into a meaning. Okay? And that's hard to do at first. I didn't have it at first, but I learned how to do this. But what helps with this, and what allows you to be able to do this, is building experience. I now have these connections, and I have these synapses working in my brain of things that I've already figured out. And now when I add more to it, guess what? It's easy because I've got a lot bigger, not bigger brain. I'm not saying that. I've got a lot bigger experience to draw from in that particular area. You know what I mean? And so learning gets easier as you go along. Like for some of you, if you stay in the health field and you stay with nursing, you finish your associate's degree. I'm not saying, I'm just saying if you do, and you move on. If you ever get to a level where you're working on like an MSN or a PhD in nursing, you will find that despite the material is so much harder, and despite you're at a higher level of learning is easier than it is right now. Okay? The hardest semesters I ever had in college were my first year, my freshman year. Okay? Learning gets easier, especially if you stick with it and form these long-term traces. Anyways, memory can be long-term or short-term. Now, short-term is going to be transient. That means it's going to be fleeting with time. They're going to form, and if you're not working to integrate as many neuronal inputs into that trace as you can to produce this image that I'm talking about, whether it's a smell, whether it's a vision, whether it's whatever it is that you had to integrate this experience to make this memory, if you're not using all the channels that you possibly can to make this memory lay down, and you're just using one, and you're using this picture of a word or this definition, you're not going to have much luck integrating a new channel. You're not going to have much luck solidifying a set of synapses that lays it down in long term. What you're going to end up with is a short-term transient memory that's you're going to stop because all it was that one thing you tried to memorize, and then when it's gone, and it's going to be gone, and you're not going to get that back. Okay. Long-term involves formation of new permanent synaptic connections. Okay, this is true learning here. This is based upon your experiences in your life and the things that you draw from. Something neat here as far as of memory. You think about amnesia, what actually happened. Now, you can't know exactly, cellularly what has happened in each case of amnesia, but in general, you have what's retrograde and anterograde amnesia. Retrograde means you can't remember anything past. Your event. Like I remember, I got knocked out one time. Remember Ryan Reichert? He knocked me out one time, and we were playing football. I was pretty young, but I got hit, and I remember laying down on the field, and my helmet was buzzing. I just remember it's like a bee flying around in there. And then all of a sudden, I had no idea what was going on. I was completely knocked out. All I remember is they told me that they carried me over to the side of the bench, and I sat down, and I remember like a half an hour later, I was awake, and I was sitting there, but I had no idea what was going on. Half an hour later, I came to, and I started asking what just happened. And I had missed out on like a half hour of the game. It was gone. I still don't. I mean, from that time, I don't have that. I can't remember. That would be sort of an example of retrograde amnesia. Especially in injuries like that are short and local. You have a half hour, whatever the duration of the event could be, you can't remember. And because the reason for that, it makes sense. These are short term. You've just formed whatever just happened. You're constantly forming new synapses, right? Everything, every word I speak, every time you glance, every time you think of a new thought, you flip the page, you're forming synaptic connections the whole time. It's a matter of, are they going to stick or are they going to unstick? Okay? Your short-term memory obviously is going to be affected, like with amnesia. They're just constantly forming, and if they're not permanent and they're not sticking and something jars it and something happens to where they just fall apart, you're not getting that back. Okay? It's just not going to happen. Another version of this is sort of anterograde, which means that you don't have the ability now to form long-term connections. This is a little more depressing. You don't have the ability to. You can remember you can remember past before. You can remember before when this event occurred. And like right now, the difference is you will always remember that, and you always remember what happened before that event. But it's to the point where. Like one example was a guy in his house. He got into a new house, and he remembered his old house perfectly well. But the problem is he can never remember where the bathroom was in his new house. He cannot form new long-term memories and only short-term transient memories. That would be sad, I think. That would be hard to watch. Did he forget his music even?
No.
Oh.
Still play. He would write things down. It was sad.
I don't know. You got to ask yourself. And I'm just thinking out loud here. You got to ask yourself about situations like that, though. It's sad for us to watch that, right? But, to think that everything was the first time that you did it. I don't know. Would it be good or bad? And he would notice and see that he's forgetting. Oh, that would be just a tormenting existence. These people are messing with you, I don't know. Anyway, I was thinking too. I'm reading a book. I study some philosophy stuff too, and it's kind of like you go around and you think about these things, but how do you know? How do you know the world was here before you were born? How do you know the world was here before you were born? What anybody can synthesize pictures. What do you base that on? Well, what evidence do you have? I mean, you have to have some sort of evidence to convince yourself of this. And if you've never thought about it, you need to. How do you know? How do you know the world was here before you were born? What kind of evidence do you use? What proof do you have? Because for all you know, the world started when you started. That's all your personal experiences are. And every person's experience is different than every other person's experience. And your set of reality is different from their set of reality because it depends on the connections that you have made. So if the only experience you have to draw from is your personal inward experiences and connections that you have made, how do you know the world was here before you were here? All right, Alzheimer's disease. Okay? Everyone is, at least to some, indirectly or something, has known someone or somebody who has had it. Maybe not personally, but it's a very sad thing. It's very hard to watch. It's one of my fears. Doing what I do for a living, the thing that would disturb me the most is if I lost my capacity to think. That would just, and it wouldn't be the point when I'm there, because then it wouldn't matter, because I wouldn't know it. It's the point leading into that where I'm realizing what's happening, and like you were talking about there, you just get so frustrated. I would hate that. Let's hope it doesn't happen. But what happens in Alzheimer's disease? The biggest problem with Alzheimer's disease right now, in terms of detection, now there's been a lot of strides made recently in terms of how to detect and diagnose Alzheimer's disease. But by and large, still what we have to come to grips with is that it's sort of like a default type of diagnosis. It's like you ruled out everything else, and this is the best thing you got. And really, even now, for sure, you don't know until an autopsy is performed and you can look at the cells that are inside the brain. Obviously, you can't do that when the person's living. Now, there's a good set of criteria. People have been working on this for a while, and it's pretty good odds that if you fit this criteria. But if we could ever figure out an actual test that's determinative, it could save. First of all, if you've got a test that's determinative, you've got something that's indicative of Alzheimer's, then you can start focusing the pharmaceuticals on whatever that which is indicative of that disease. So if we ever get to that point, early detection becomes extremely important, because then hopefully you can medicate it or whatever. There's some real good Alzheimer's research just going on down the road in Dr. Young's lab in the Terre Haute Medical Center for Medical Education on the ISU's campus. But anyway, Alzheimer's disease, what's characteristic of it is what's called amyloid plaques. You get these actual plaques that develop. There's where the endings are. These nerve endings start to degenerate, and there's almost like little callus-like protein fiber kind of things towards the end of these endings. And what happens is they actually sort of form like calluses and plaques inside of that, which significantly impedes neurotransmission through that axon. Not only that, but up at the top of that axon towards the cell body, you have what's called neurofibrillary tangles. Similar things sort of just get all mushed up and tangled up. These sort of protein filament type things that are in the cell bodies. I don't understand exactly what causes it, where it's all coming from. There's certainly some proteins that have been identified and pathways that have been identified, but no breakthrough stuff yet. But what happens is basically as these neurons cease to function, they cannot transmit those impulses like they once could, and thus you're going to lose memory traces when that happens. And it's, I don't know. All right, I mentioned the brainstem a while back. Here's a picture of why it's so important in terms of integrating everything. Here's a bunch of the cranial nerves. You don't have to know all of these, but notice where they all come from. Right in that region, right around the base of the brain, the brainstem region. So as far as integrating sensory input from our body, extremely important. That's the hub. That's where it all comes through. Okay. Your basic functions associated with life. Your breathing, your respiration patterns, all this is going to be controlled by regions of the brainstem. Now, sleep. This is pretty interesting. The thing is, sleep is actually a very active process by our bodies. And you could actually, use as much energy and have as much going on physiologically, if not more, when you're sleeping than when you're awake. So what then is the point of sleep? Because if you're looking at the biology of things and you're looking at the physiology, you're not resting. You're actually still working as if you were awake. So why do we sleep? It's interesting. First, though, let's consider what we call the 4 levels of arousal. Okay, the 4 levels of being awake. You've got worst case scenario, coma, right? And then a step up from coma would be being asleep, and then sort of wakefulness is where you're about right now. And then maximum alertness. Okay, so you've got about 4 different levels there. So I point out here that sleep does not reduce brain activity. If you look at the EEGs, for example, you will see that activities can even increase in the brain. Sometimes oxygen use even increases. You actually sometimes suck in more oxygen when you're asleep than you do when you're awake. We've got 2 different stages, what we'll call stages of sleep. We've got what's called slow wave sleep and paradoxical sleep. And it's going to be in reference to the EEGs that are associated with these stages of sleep. And this is going to explain some things in terms of, when you wake up at a certain time from when you went to sleep, why is it that you're more tired? Sometimes you could get just as much, some days you're more tired than others. Why is that the case? Why is it that you can, take a 20-minute nap, you can take a 40-minute nap, and you're more tired when you wake up from the 40-minute nap as opposed to the 20-minute nap? Okay? So take a look at some of this. Slow wave sleep, within it, actually has 4 stages within that stage. Okay? We don't have to know all of those, but the point is, this lasts for about 30 to 45 minutes and then reverses. In other words, you have a stage of your sleep that you'll go through. You can monitor this on the EEG, that you'll go through what's called slow wave sleep. And from the time that it starts, it'll go through, and it'll peak at about 35 to 40 minutes, and then it'll reverse itself down through that pattern. Okay? So it goes up, it goes back down. Now, what is happening during a 30 to 45-minute phase of what we call slow wave sleep? Your body is maintaining muscle tone. Okay, just kind of like you're sitting here now. You're maintaining this muscle tone. Your body can be shifting. You shift a lot while you sleep, some more than others. Dreaming is very rare in this, anywhere from an hour to hour and 15-minute, hour and a half interval, whatever this is going to be. Dreaming is very rare because you know that you don't dream all night long, right? There's actually very, very brief moments that when dreaming occurs. Is dreaming reality? That's the question. Because, if you define reality by your experiences and synaptic connections, that's the same thing that's happening when you're dreaming. So how do you say that one is real and one is not real whenever they involve the exact same physiological processes and the exact same things are happening? But you've defined that norm. You've defined that norm. In your head, you've got synaptic connections saying that clothes are necessary. We're not born with them, but somehow they're necessary, right? You have yourselves, I've got this quote that I like to say. No one will ever quote me because I've never written anything, but this is my quote. The greatest tragedy lies with those who think that the believable must be logical because we ourselves define the parameters of logic. In other words, we decide what the box is in which we want to think. We define that. We say this is normal, this is not normal. This is how these are the laws I'm going to use, these are the data I'm going to use, this is how I'm going to think about this, and we don't allow ourselves to think outside the box. You've heard of that before. So the fact that you say, because I don't have any clothes on, because I'm flying on an elephant, and so I know it's a dream. Well, my question is, how do you know that's a dream? How do you know? How do you know? You have no idea. You got no proof? Show me some evidence. All right. All right. But what I will mention here is that dreaming is very rare, but nightmares are not. Nightmares can happen in this phase of sleep. Okay? I'm separating dreams and nightmares as positive, negative experience types of things. Okay? Now, the thoughts that you would have in this stage of sleep are more conceptual and more plausible, which is interesting because which one feels like you're there more, a dream or a nightmare? Yeah. You can feel your heart rate increasing. You can feel this stuff like you're actually there when you're experiencing a nightmare as opposed to floating on a cloud on a magic carpet somewhere. Okay? So it is interesting that this is where nightmares seem to come about. And the thoughts associated with them are more conceptual, more plausible. That doesn't mean they're realistic necessarily, but they are more plausible. They're more likely to be believed, for example, than, at least it feels like it to you, than some of the other things, some of the other types of thoughts that you could have. Now, there's another stage of sleep called paradoxical sleep. Paradoxical means it's sort of, it doesn't fit the mold, it kind of goes outside the box kind of thing. About 20% of your sleeping time, it's only about a fifth of your sleeping time is spent in paradoxical sleep, whereas the rest of it's in this slow wave sleep. So you go slow wave, reverts back through that same process of slow wave, paradoxical sleep. Slow wave, paradoxical sleep. You're cycling through this. Okay? Only lasts about 10 to 15 minutes after the slow wave. Okay? All right, now if you look at a person's EEG when they're sleeping and they're in the stage called paradoxical sleep, it looks like they're wide awake. It looks like everything is functioning like they would be awake. There's no muscle tone, however. you've lost your muscle tone at this point. You have high REM, high rapid eye movement. Now, people used to think that rapid eye movement was associated with the dreaming process. Now that's shown to be not true. They're not sure why rapid eye movement occurs, why you flutter your eyes whenever you sleep. People would sort of liken it to like a cinematic kind of picture where it was flipping up and down and the picture was being displayed on your eyelids and that was your dream or whatever. That was a long time ago. But there is no, the dreaming that has been recorded tends to show no relationship with the actual REM itself. So they're still trying to figure out what the function of rapid eye movement is because it's interesting. There's really, can't understand why it would happen. Back up one, irregular vitals. Your vitals are all over the place when this happens, okay, in this particular area of sleep. Dreaming really stimulates the visual and limbic system. Emotions, especially those of happiness, sadness, not just fear, not just biological survival type of things, right? When you think of fear, you think of fight or flight, you think of situations that are essential to survival, very basic instinct type stuff that are much more conceptual, much more plausible. Okay? Down here we're talking about visual limbic systems. That area of your brain tends to be more excited. That contributes to the thoughts that you're having in this the synaptic activity that is occurring in this stage of sleep. You've got reduced function in the reasoning center of your cortex, which makes sense and why some of these dreams that some people may have are way out there, because you've actually decreased your ability. Well, you haven't done anything, but by virtue of the fact that you're in paradox sleep cycle, you have reduced function, you have reduced reasoning capacity. That area of your brain works less than what it normally would. As to a reason, I don't know why. No one really does. This is just the things that we have noticed. Well, I haven't. The researchers have noticed. Infants tend to spend more time, that's something interesting, they tend to spend more time in paradoxical sleep than they do up here in slow wave sleep. Who knows? What is a suggestion as to why that occurs? It looks like, based upon what's going on here, there's a lot more synaptic firing that's occurring. So does that contribute to the synaptic connections that babies are trying to make in terms of growing from experience and the thoughts that they are having and constantly trying to create more long-term type of synapses? And we do know that the baby's brain is extremely plastic, right? It can assimilate information like crazy, much faster than any of us ever could. The function of sleep is very unclear. Here's the leading theory. I put this one up here. There's other theories, but this is the one that seems to have the most credibility so far, is that there's a buildup of extracellular adenosine when you're awake. Remember, adenosine is related to adenosine triphosphate and all that kind of stuff. So through the metabolic activities normally of these cells, there tends to be a buildup of adenosine on the outside. Now, when this gets on the outside of the brain, in the brain tissue outside of their cells, within the brain, in that extracellular space, it has to be dealt with. And there's a cycle. The amount of adenosine continues to increase to a certain level, and then we go to sleep. And then while we go to sleep, it tends to sort of absorb and pull that adenosine back up and get rid of it and get it out of the ECF and brought back into the places it should be. Perhaps that is the sole function of sleep, to deal with that. Well, insomnia is going to has to do with, well, I'll talk about that in a second, but not insomnia especially, but the sleep-wake center in your brain. There's a part of your brainstem that's actually controls these cycles. Okay? So if you have insomnia where you actually cannot sleep, then you've probably got something just wrong with the control center that's controlling these cycles. It's not, it's just an irregular pattern or whatever. Now, as far as like sleepwalking, and something like that, I don't know near enough about it to answer that question, but you can see where it's somehow there's a merger probably of this, what's going on here with your cycles. You start taking a phase of sleep where you have muscle tone and your body is able to move, but yet, cognitively you're down here somewhere where you can't really, you're thinking sort of weird things. You're thinking you're walking off a cliff somewhere, but really you're going to the kitchen, something like that. It just depends. There's probably some research out there, but I'm sure it's incredibly complex, and I've not read it. But it would be interesting. Are you asking, can you, well, what I'm saying is, let's see how I can answer your question because I think I know what you're asking. There's something called circadian rhythms, and there's something, and then you've got these actual sleep cycles. The difference is when you go to sleep, what cycles do you go through? These. When you decide to go to sleep, what biological controls make you tired? There's a lot involved. What reactions are going on in your cell throughout the day to cause you to get these cycles started and get you to go to sleep? Realize that these don't control when you go to sleep as much as they do while you are sleeping, so that you can sort of keep the two separate. And I'm going to call one of those circadian rhythm, okay, which is what you're talking about. You can affect your circadian rhythms to a degree. You can, get on a different time. You can put your body, like if you're working on nights versus working on days, you can, over a period of time, actually cause your body to shift. But what you'll see is, regardless of whether you sleep during the day or whether you sleep during the night, you still go through these same stages. Does that make sense? I didn't do it very well, but I think I got the point across. All right. Well, anyways. Let's see. Oh, this is interesting, too. Why is it that you can drink a lot of caffeine, for example? Why does that keep you awake? The neuromodulator adenosine, I told you, that's present, whenever adenosine starts to build up, okay, and it starts to build up extracellularly, what it does is it starts to work biochemically against what's the arousal center. Naturally, you have a center in your brain that causes you to be aroused, okay, to be awake and conscious of your surroundings. So the cycle, as it works, and as your adenosine levels are building up outside of your cells, what tends to happen is the more of it is there, it starts to inactivate. That's why it's a gradual process. You don't walk and well, some people do, but you usually don't walk and all of a sudden you're not asleep. Okay, it's a gradual build-up process. So as this adenosine continues to build, it's activating less and less your arousal center. Okay, so it's working against arousal, and the more adenosine, eventually it gets to the point where you go to sleep. Okay, now caffeine actually blocks the adenosine receptors on this arousal center so that adenosine cannot come there and activate it and cause you to get tired, basically. So the fact there's a chemical interaction there. The fact that you have caffeine present, it gets on those receptors on the arousal center and prevents adenosine from binding to it. So if adenosine can't bind to it, you're not going to get tired. Okay? I'm not saying that's a good thing. I'm just saying that's what happens. All right. All right. So if you look at slow wave sleep versus paradoxical sleep and then compare that to what you look like when your eyes are wide open, it's pretty interesting. Your slow wave sleep is actually that long stage. Okay? You have muscle tone, you have nightmares, you have fairly regular vitals and that kind of thing. But for some reason, you're sort of spastic, the waves are much bigger, it looks very different. Down here, in your paradoxical sleep, it almost mimics what it would be like to be wide awake and open. So are you awake? Are you experiencing a different reality? Have you transgressed into an alternate universe at that point? It's good stuff, man. It's good stuff. Okay, real quick. That's just you don't have to know any of this. You know what the spinal cord looks like, where the regions are. There's tons of branches coming off this. This is very, very dense with axons especially. A lot of neural tissue in here. You have ascending tracts and descending tracts of the spinal cord. What I'll point out here, I'm going to show you a reflex arc here in a second, but if you have a feed coming in, an afferent feed going into the central nervous system, notice this gray area. So you've got some cell body type of area here, some darker matter. You've got some dense axonal matter out here from these neurons. But the point is, you've got these interneurons that are inside the central nervous system that connect afferent and efferent pathways, okay, coming in and out of that spinal cord. In the case of a reflex, what you will see is you've actually got some feeds that will come into the spinal cord that will just pull this U-turn just like this, fire that one, and feed right out to like a muscle, for example, to pull something back. Where at the same time, this neuron right here may branch off and connect to another one and send it straight up to the brain all at the same time. So you've got a reflex that you don't have to think about, but while you're not thinking about it, then you think about it. Okay, if that makes sense. It's two separate tracks where that occurs. So it's a control mechanism in that sense. So there's an example of what I'm talking about. If you take a look at this is interesting too, I want to point out. You touch something hot, you pull it back, right? Okay? You've got a feed, an afferent feed coming in here. Notice the interplay that is happening in here. You've got a couple different, you've got an excitatory and you've got an inhibitory interneuron here. This is where it all is going to come together. This excitatory, this particular feed would excite this, and this synapse is going to be an excitatory synapse. So what it will do is feed over here and cause your bicep to contract, which will pull your arm up. Now realize the only way your arm is going to be able to pull up, these are antagonists, your bicep and your tricep. So the only way this bicep is going to be able to contract and pull your arm up is if the other one relaxes, right? So there has to be an inhibitory message sent. At the same time, notice the same neuron branches out and connects to this one and forms an inhibitory connection. That fires, comes over here, relaxes the tricep all at the same time so the bicep can contract. Now also at the same time, notice what's happening. Another branch of this same input neuron relays, goes over here to the right side. Notice that it goes all the way over to the white matter. That's where the axons are. Okay? It actually crosses the spinal cord. It goes from, let's say, the left to the right side, and then there's an ascending pathway. That goes up to the brain for you to cognitively process that which has just happened. So yes, it is true that the right side of your brain especially would control what's going on over here. And the left side will control what's going on over here. And it actually occurs all the way down here at this level. You get a relay across. I'm not... Yes? Well, okay. Well, okay. I'll look at it here in a second. Just ask me. I know we're about. Hang on a second. I want to try to fly through this. This is a picture of a nerve. Realize what a nerve is. It's a whole bunch of axons all bundled together. Okay. Same thing here. Know the parts. The afferent pathway. The efferent pathway. Remember the efferent pathway. What it's going to land on is something called an effector. An effector is going to cause an effect to happen. It's going to be like a muscle, for example, that will do that. You should be able to figure this out yourself. You should know that you have a receptor, an afferent pathway, an integrating center, an efferent pathway, and an effector. You can go through those on your own.